we're going to talk about the mechanisms and treatments of schizophrenia. So the first thing we need to talk about are action potentials. And action potentials are a change in voltage across the cellular membrane to propagate an electrical signal, which will eventually lead to the release of neurotransmitters, and this will cause more action potentials to be caused, I guess, in the neuron. So how exactly is the action potential generated? So first we have our resting state, and what we have is in the extracellular part of the membrane, it's mostly positive, and this is due to a high concentration of Na plus outside the membrane, so plus, 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 plus. And then on the intracellular part of the membrane, there's a lot of K plus out here, or in here, sorry, but not as much as Na plus here, so, and there's not a lot of K plus outside here, so maybe like one charge. And so in a resting state, we have these sodium channels which are closed right now, so they're closed. And so what will happen in action potential is so we have our receptor, and we have a presynaptic neuron. And the presynaptic neuron, after its action potential finishes, it'll cause the release of these neurotransmitters. And these neurotransmitters will actually bind to the receptor, and the binding of the receptor will cause these Na plus channels to open. So they're open right here, open. And since there's more in the resting state, there was more Na plus here, so a lot of Na plus here, and then there's small Na plus inside the cell. It'll cause the Na plus to flow down its concentration gradient, so it's heavily flowing. This is called the depolarization of the cell, because now, right here, the depolarization of the cell means there's now a lot of positive charges inside here, and the extracellular part of the cell is kind of negative. So as more Na plus goes inside the cell, the membrane potential rises. So as you can see in this graph right here, it's rising, right, rising, and then it reaches a certain point where these channels close. So now no more Na plus can enter the cell. So it's kind of in this weird equilibrium state. But now the K plus channels open, and as we recall, there's in the resting membrane, there's a lot of K plus here, and then little K plus, but as the potential changes, I guess, the K plus begins to flow down its concentration gradient, right? And this makes the cellular membrane to become negative again. So this can kind of be seen right here in this graph. So it's falling as the cellular membrane becomes more negative. So then kind of returns to the resting membrane potential right here. And so what happens is this K plus is still kind of flowing in, but then the K plus channels close again, and it returns to the resting membrane potential right here. So some neurotransmitters are able to cause excitatory or inhibitory effects. We're going to talk about excitatory effects first. So excitatory neurotransmitters increase the frequency, or maybe not frequency, increase the likelihood that a neuron will fire an action potential. So this is what an excitatory action potential would look like on a graph. So we usually have a threshold action, or threshold membrane potential that the action potential kind of needs to reach. So in excitatory, right, we reach the threshold and we reach the top, right, and this causes the release of, or the generation of, an action potential. Then in an inhibitory neurotransmitter does the exact opposite. It'll decrease the likelihood that action potential will happen. And this is done because the action potential never reaches this threshold. So what an inhibitory neurotransmitter might do is it'll cause it maybe to slowly fall down, and it never maybe even make the membrane potential even more negative, and then it'll go back up, right, to repolarize, and it never reaches a threshold, so an action potential is never generated. So what are important neurotransmitters that for schizophrenia? So glutamate is an important neurotransmitter here. Dopamine, and that's excitatory, inhibitory, and serotonin, which is mostly inhibitory and excitatory. And this will come into play when we see drug effects actually acting on these neurotransmitters and receptors, which is kind of cool. So here are the structures of neurotransmitters. Again, here's dopamine, here's serotonin, and here's glutamate. So we're going to talk about the symptoms of schizophrenia, and it can be separated into positive and negative symptoms. And negative symptoms are kind of the lack of ability to function normally in society. So some positive symptoms include delusions, hallucinations, disorganized thinking, and disorganized or abnormal motor behavior. So the negative symptoms are kind of a flattened effect. They'll have a monotone flat speech. They'll have a loss of interest in certain activities, and they'll be socially withdrawn. So we're going to talk about the different causes of schizophrenia. And so there's three main hypotheses or causes, and that's the dopaminergic hypothesis, the glutamatergic hypothesis, and the serotonergic hypothesis. So we're going to talk about the dopaminergic hypothesis first. And so basically there's two problems that are happening with dopamine. So in the mesolimbic pathway, dopamine, there's too much dopamine binding to the D2 receptor, so it's severely binding to this D2 receptor. And then the problem with the mesocortical pathway is that not a lot of dopamine is being binded to the D1 receptor, so not a lot of dopamine binding there. We're going to talk about the serotonergic hypothesis, and what this states is that there's too much serotonin binding to the 5-HT2A receptor, so too much serotonin binding to the 5-HT2A receptor here. And then the next hypothesis is the glutamatergic hypothesis, and glutamate is the main excitatory neurotransmitter. And the problem here is that there's not enough glutamate binding to the NMDA receptor. So, not enough glutamate binding to the NMDA receptor. So we're going to talk about the different drug treatments of schizophrenia. And so we're going to talk about the first generation drugs and haloperidol. So this is just the structure of haloperidol. So as I stated before, one of the problems with schizophrenia is that there's too much dopamine right here binding to the D2 receptor. So our solution to this problem is we want to decrease the binding of dopamine to the D2 receptor. So decrease it. And that's what haloperidol does. It competes with dopamine for binding at the D2 receptor and decreases the amount of dopamine that can bind to the D2 receptor. And this is just known as antagonism. So this is just antagonism, basically. So we're going to talk about the second generation drugs, specifically clozapine, risperidone, and aripiprazole. And so this is just the structure of clozapine, this is the structure of risperidone, and this is the structure of aripiprazole. So as I stated before, right, there's too much dopamine binding to the D2 receptor, and there's also too much serotonin binding to its receptor at the 5-HT2A receptor. So what we need to do is we need to reduce the activity at these receptors, right? So decrease the amount, again, at these receptors. we need antagonistic activity, and that's exactly what these drugs do. Is they compete with dopamine, serotonin for binding, and decrease the amount of dopamine and serotonin at these receptors. So the main drugs that are being looked at, it's called ITI-007. And so, like the first and second generation drugs, it acts as an antagonist for dopamine and serotonin, so antagonist. But it also helps to increase the amount of glutamate at the NMDA receptor. So, as I stated before, there's not enough glutamate binding to the NMDA receptor. So, ITI-007 actually indirectly increases the amount of glutamate binding to the NMDA receptor, and this is called agonist activity because it's increasing the amount that the neurotransmitter will bind to this receptor. Those are the current drug treatments, and those are the actual current hypothesis and core causes of schizophrenia. And these are all my references, and yep, that's it.